Yeah. So yeah, again, this is our second physics seminar of this semester. So again, we're trying to do everything on Zoom this semester. So most of our speakers are from a different location, and but this week we have our second speaker. I mean, Dr. Nathan Green. He's from NSU, but he's based in at Broken Arrow campus, and so again, it saved him a little bit travel to Tahlequah, and so that he can give his talk in his office. So again, it's our pleasure to have him with us. Today, he's going to talk about nanoparticle assemblies. So go ahead, Nathan.
All right. Well, thank you very much for having me. And I'm sorry this talk was actually delayed by a year. I was supposed to give this talk last year, and then I got sick before COVID. So it's been tough for me to make it in the physics seminar, but I'm very grateful to be here, and I'm honored to follow up Ralph Layton. You know, I'm not on the same playing field there, but I'm a huge Feynman fan. So Dr. Zhang, good job on getting a good set of speakers together, because that really was a treat to watch after the fact. I'd like to talk to everybody today about the work that goes on in my lab on the Broken Arrow campus, where we are in the business of making and assembling nanoparticles. And it turns out that there's some tricks that we have to play because the particles are particularly small. So let's see. There we go. So what we'll do is talk a little bit about nanoparticles in history. They have a pretty neat historical context. What nanoparticles, the structures that we're looking for, how to make those structures, functionalize them, and put them together, and in particular using this DNA-based method called DNA origami, and then what we'd like to do with those. And so I'll talk about some of the physics of how we're hoping to use these gold nanoparticle aggregates as energy transfer modalities. But before I get into all of that, let me tell you about some of the neat things of how nanoparticles showed up in history and we didn't even know it. So as early as the 4th century, but I think even earlier than that, nanoparticles had been used in a lot of glass making, but we weren't aware that that's what was going on. There's this cup, the Lycurgus cup, and I believe this is in the British Museum of History right now. That is kind of this green color, and actually, can you guys see my cursor if I use a mouse?
Yes, I can see it.
So it's got this kind of green color when you have light shining on it, but when you shine light through it, so this is backlit, you can see that the cup looks red. And there was a couple of these cups that were famous for this. They were thought to be kind of enchanted, and so they were meant to be for like very special guests of honor. What's going on here is that there are little nanoparticles that are embedded in this glass, and in this case, they're gold nanospheres, and gold that I'll show you here in a second, when it's particularly small, you can change the wavelength of light that it absorbs, and so you get this nice red color. Another place where you would see that, maybe this looks kind of ugly, but this looks quite nice. Another place that you might see that red sort of color, and here's a TEM image of particles that are a thin section of that glass. You can see the particles that are stuck in there, is stained glass. So modern stained glass doesn't have nanoparticles in it, but way back in the day, whenever you wanted to make really beautiful stained glass windows, what you would do is pour in these metal salts into the molten glass along with some reducing agents. And what would happen is the gold salts, which were anywhere from, or it doesn't have to be gold, but we'll talk about gold here. It could be a gold 3 salt. As the reducing agent interacts with the gold in the glass, it turns it from gold 3 to gold 2 to gold 1, those are ions, to gold metal. And then as the gold metal starts to form, it creates nanoparticles. And so you can see different collections of those. And sometimes they're spheres, sometimes they're triangles or cubes. Often they are symmetric though, because nature tends to like to grow things symmetrically. It's the lowest energy pathway to doing that. And the particles would get suspended in the glass so that you start to see, you can make different colors by using gold nanoparticles of different sizes. You can get reds to yellows and oranges. If you put silver nanoparticles in there, yellows and blues and browns. And so a lot of, if you go to Europe especially, a lot of the stained glass windows that were put in that are original are really just glass full of metal noble nanoparticles. And so we didn't know that for a long time because these particles are so small that you can't actually see them unless you have an electron microscope. We were good at making glass, but we didn't really have a source of electricity or vacuum strong enough to make an electron microscope. So it wasn't until much later on we found out what was going on here. A couple other places that you see gold nanoparticles show up that are interesting are on these lateral flow assay tests. So the way this can work is you have something like a pregnancy test where there's a wicking pad on an end where you put a sample, and then the sample flows across an area where there are antibodies labeled with some sort of fluorophore. And a fluorophore in chemistry terms is something that's colored or something that fluoresces. This can be gold nanoparticles. And so what happens is if you have an analyte present, so for pregnancy tests, it's HCG, that'll bind to the antibody and carry it over to two different strips. One of the strips has the antibodies present without the analyte. And then the other strip, this one actually, this graphic's from NASA, and so this one really should have the analyte there. That would be HCG molecules that stay put, so that the gold nanoparticles would line up here if there was HCG present, and they would absolutely line up here just to show that the test works. The way that you would normally see this on a pregnancy test are these two lines. And so one line is the control line. This red color comes from the gold nanoparticles. They have bound along the control line, and then here's the test line. And the two lines would indicate that a pregnancy was detected. That gold nanoparticles are a great fluorophore for this test because you can imagine a person would want to know. They want this test to be accurate. And so if you use dye, you have to worry about, does the dye photo bleach or go away so that the color isn't recognizable anymore? And if that's the case, then you'd get false negatives, which is obviously a big deal. Gold nanoparticles don't do that though. You can leave gold nanoparticles in the sun as long as you want. And this red color, this is an image from my lab. This red color persists basically forever. You can't do any chemistry to them that's going to make the red go away. Sometimes you'll also see blue lines on pregnancy tests. Those come from latex nanoparticles, but either case, you want a material that is indelible, that cannot lose its color, so that you can trust the test. And that's why you have the two lines as well, is to make sure that the test is actually working. So it's kind of odd to think about when I'm saying, oh yeah, these are gold. You probably are wondering, well, isn't gold colored? You know, we know gold has a really shiny yellowish look to it. That's true on the bulk scale, but when you shrink a gold nanoparticle down small enough, it becomes colored. And so this is chloroauric acid. You don't need to know the molecular formula for it, but it's gold 3 in here. And it has kind of a goldish color. But as we reduce that chloroauric acid, gold 3, into gold 0, it turns into nanoparticles, and you can see that red color. And I put these side by side so you can hopefully see the red color shared across these pregnancy tests. Now, that doesn't mean that we need to go and buy up pregnancy tests and get the gold out of them. Very small amount of gold nanoparticles are very highly colored, and so the amount of material you need is negligible, but it's mainly because the color stays so well. So why is gold not gold? Why is gold red or blue or any of these other colors I was talking about? Well, it comes from the fact that when you take a gold solution and you turn it into a nanoparticle, these nanoparticles are smaller than a wavelength of light. And so here you can see this is a TEM, that stands for transmission electron microscopy. We have to use electrons because our particles are smaller than a wavelength of visible light, so you can't see them with visible light. So you can see the structure here with these electrons. This is a cartoon of how the gold nanoparticle would interact with a wavelength of light. So it's a metal nanoparticle, it's electron rich. If you made a wire out of gold, it would conduct electricity very well with very low resistance because of these valence, or what we call conduction band electrons, surround the outside of this particle. So a wavelength of light is really two orthogonal fields of electromagnetic radiation. Well, the electric part of this, as the wavelength goes over top of the particle, would push that electron cloud down, and as the wavelength comes underneath the particle, would push the electron cloud up. And so that cloud goes down and up and down and up and down and up, and it resonates back and forth. Well, in chemistry terms, when we talk about something resonating or a dipole resonating, that's really absorbing light. And based on the size of the particle, you can change the wavelength of light that you absorb. And so for most spheres that are sub-nanometer, that are sub-visible wavelength range, so these are about 20 to 30 nanometers, depending on where you measure them, they tend to look red. Now I'll show you some gold nano rods later on, and we can make those blue and other colors based on the shape. But we call this the surface plasmon resonance. Plasmon is a collection of electrons, a cloud of electrons, and the surface, they're bound to the surface, and then resonance, they're kind of bouncing back and forth across the particle. Now, importantly, in a sphere, the resonance is equal. I believe, and I try not to mess up the physics here too much, but I believe that there is a physics term used for this called the flat Earth principle or something like that. And it's not flat Earth, but if you took every landmass on the planet and you plowed it to where the oceans covered the surface of the planet evenly, so basically the entire surface of the world is water, and you hit the water, you hit the Earth with something heavy, the water would slosh back and forth across the surface of the globe because it's still being held to the surface with gravity. That's the same thing that would happen here. You can envision the electrons around my metal nanoparticle as water, and it just sloshes back and forth to a certain frequency. And whatever frequency that is, that's the frequency of light that it'll absorb. So for most of nanoparticles, it's absorbing a lot of blue light, which means it reflects back red light, and they look red to us. It's more than you'd ever want to know about gold nanoparticles. So why is that interesting for us? If you can absorb light, then we're interested in what can you do with that light? So we can put energy into a gold nanoparticle. Can we do anything to get the energy back out? And one system that we've been looking at in my lab for quite some time and following on from my graduate work is, can we get fluorophores to interact with metal nanoparticles? And so what we did is we took a fluorophore, which is, now I'm using the word fluorophore specifically as an organic molecule that fluoresces. So it takes in light, goes to an excited state, relaxes vibrationally, and then gives off light of a different wavelength. So there's this this lag between the light it absorbs and the light it gives off. We wanted to put fluorophores inside silica because it turns out if an organic dye or fluorophore gets right on the surface of gold, it quenches, it just turns off. So when we put it in a silica marble, it provides a spacer for us. And the silica also provides us a place that we can put binding units to the outside. So then I can bring in my gold nanorods and have the gold interact with the fluorophore. And it's our hypothesis, or it was our hypothesis, that if we did this on a small scale and surrounded enough dye-doped silica nanoparticles, or fluorophore-doped silica nanoparticles, with gold nanoparticles, that we could get the fluorophores to glow much brighter. And so we set out to do this through a couple neat chemistry tricks. I won't talk about how the silica is made, but it's a pretty cool process. And actually, let me just glaze over this. This is good for the chemists in the audience, but since we're physicists here, we're interested just in what's going to make these things glow brighter. We do a quick transformation. This group on the end is called an amine. This is a primary amine. When you react that with carbon disulfide, sorry, you can turn this into something called a dithiocarbamate. And you don't need to know chemistry for this, but sulfur loves gold. For whatever reason, they stick together really well. This is cement. So whenever we make these two sulfurs that share a negative charge, they love gold nanoparticles. And so those are the green cups that I was showing you on the previous thing. When you put those together and then mix in gold nanoparticles, we get these aggregates to glow very brightly. And so these are a couple electron microscopy images to show that we were able to get these aggregations to occur. This is a TEM, so a transmission electron microscope. That works by having a thin sample that you pass the electrons through, and so you see a shadow on the bottom of it. This is an SEM, a scanning electron microscope. That works a little bit differently, where you don't send the electrons all the way through the particles. You let them absorb into the surface of the sample, and then they eject electrons back, and so you're reading them. And it gives it this kind of three-dimensional look. So here, the silicon nanoparticles are kind of a lighter color. They don't absorb the electron beam as well. The gold nanoparticles are darker in color. They have a denser nucleus, so they stop the electron beam better. And so the opposite is true here. The gold looks lighter because it returns more electrons than the silica does. So the silica has kind of a gray look to it, and the gold has kind of a white look to it. And we can get quantitative with this to show it, but I'll just trust me here that there's gold all over these silicon nanoparticles. So I was really excited to get this image. It's actually the thing that allowed me to finish my dissertation on time for making these aggregates. And not only can we make them, but it turns out that they work appropriately. Here's a different view of that, showing how the gold really lights up when you look at one particular source from the electron beam. So when we had our dye-doped silicon nanoparticles, this is a fluorescence intensity emission scale. So basically, you can't see them on this scale if there's no gold present. When you add gold and you let them mix for a while and those aggregates start to form, this is the same solution, just capped, taken over a series of days. After the first day, we went from basically no emission up to just below 800. After 5 days, above 800. And after 7 days, we actually couldn't measure it because the intensity got so high that it maxed out our instrument here. So we saw this tremendous increase in fluorescence intensity. And there's a lot of reasons behind this, and I'll be happy to talk about it during the question and answer if you'd like. But to oversimplify it, the gold and the fluorophores get together and provide another extinction pathway so that when the fluorophore absorbs light, it hands it off to the gold and then emits. And it allows the fluorophore to turn over faster. So something we call the quantum yield, the ability to take in light and give it back out, is increased quite a bit. And here we were able to show that it does matter. It's not just the gold and silica being in solution. So this is a really zoomed in scale. You can see here, SINP means silicon nanoparticle and GNP means gold nanoparticle. If you have just silica and gold in the solution, that red line, it doesn't work. If there's no glue, no dithiocarbamate to hold them together, you don't see any increase in the emission. And if you have just silicon nanoparticles and no gold nanoparticles, same story, no increase in emission. So we were pretty excited by this. And work is still ongoing at OU following along these lines. But something that always stuck with me about this was, as neat as these slides are, they're pretty messy. And I'm just like arbitrarily, it's like there's like ticks all over these silica. And I didn't really get to engineer them very much. And so we have always been interested in working with gold nanorods. And inherently, when you work with a gold nanorod versus a sphere, you're working with an anisotropic structure, meaning that you could put particles in the ends versus the sides, and it would be different. And so we wanted to start looking at gold nanorods and how we could better assemble these molecules to try and get similar properties, but maybe a little more elegant. So it's pretty easy still to grow a gold nanorod. It's not as easy as a nanoparticle because you have to grow them long, but through a series of chemical tricks using a surfactant called CTAB and silver, we can build this kind of soap tube that the gold nanoparticle can grow in. And so we put in a seed solution and then feed it gold slowly. And what that allows us to do is to grow these elongated pill form gold nanorods. And you can see they form pretty well. They're discrete. They're all about the same size. We call that monodisperse, all about the same size and shape. And the coolest part about them is they are tunable. So with a gold nanorod, that plasmon I was talking about, the vibration of electron density, can happen in two different modes. So with a sphere, it's just equal all around the outside. But with a rod, it can go along the long axis, what we call the longitudinal, or it can go across the short axis, what we call the transverse plasmon. So this peak here, about 500, represents the short transverse axis. And then these peaks are the longitudinal. And the part that's neat here is you can see the aspect ratio, that is the length per width division. So as the nanoparticle as the nanorod gets longer, the aspect ratio goes up. You can tune where the longitudinal plasmon absorbs. And this would allow us then to play with what kind of dyes we can put in our silicon nanoparticles to see how the energy transfers between the two. And so in my lab, we like to make particles that are around 630 nanometers, even shorter than 2.4. And those solutions tend to be blue. So I will, to the best of my ability in here, give credit to my students who have pioneered this for me. Stephen Kane was an undergrad who really helped set up my lab when I first got here about 4 years ago. And he came up with the process we were using, that we still use to this day, to make gold nanorods. And so you can see here the seed solution that I was talking about has kind of a reddish color. Those are really small particles, and so they look kind of red. As you feed the seed solution into the growth solution, the spheres start to turn into rods, and you can see this second peak start to develop. So this is the transverse peak, and this is the longitudinal peak. He was making a concentration curve here to figure out the concentration. So that's why these are just increasingly concentrated samples of the same gold nanorod solution. But you can see this nice blue that you get from this. That's when you have a longitudinal plasmon about 630 or so, you see this blue solution. So it's always really obvious when we make the particles well, because it's really hard to make gold look like it's blue. But we've gotten quite good at it. So what do we want to do with them? Why are we talking about rods versus spheres? With the spheres, it was interesting to see if we could get a bunch of silica spheres and gold spheres together. With a rod, there is an opportunity here to take advantage of the anisotropic formation of it. And what we'd like to do is see if we can get the energy back out of a gold nanorod. So about 10 years ago, there were some pretty convincing experiments that energy can go into a gold nanorod. A lab actually took a really fine point laser and shined it on a gold nanorod and showed that the nanorod lit up, that it absorbed the energy. But what we are not sure about is, can we get the energy back out of a gold nanorod on the other side? Kind of like a fiber optic cable. And so what we'd like to do is take a di-doped silicon nanoparticle and attach it to one side of a gold nanorod, and then take a different di-doped silicon nanoparticle and put it on the other side of the gold nanorod. We would then excite the donor di-doped silicon nanoparticle, so this guy on the left, see if it could transfer the energy into the gold nanorod along that plasmon, and then transfer the energy to an acceptor di-doped silicon nanoparticle on the other side. So we put light in here and get light out over here. And I've chose these colors carefully. So this being a little more blue, violet, purple, higher intensity light, this being more red, lower intensity light, because that's typically how a Stokes shift or the decrease in energy from absorbing and fluorescing and emitting light, how that tends to go. And so why we'd be interested in this, one is just, can we build something that does this? Are gold nanorods capable of it? But there's also some really interesting applications for solar cells. One of which being, and this is going to be one of these things you guys have probably picked up on with basic researchers, is that we will sell lots of applications and then we're like, but we would never do this work. So one of the ways that this could work would be to take our nanoparticle complexes and put them into a solar cell so that you have one side of this assembly facing sunlight and another side of this assembly facing where the electron ejection would occur. And what that would allow us to do is separate those two processes across a larger area. So right now the way it works is you basically take silicon and slap them together in what's called a p-n junction. And you put a charge on one side, you put a charge on the other side, and hope the electron jumps across from one side to the next and then creates a current. Well, that happens, but what also happens is that the charge is created and then recombines, and all you get is heat. And so our solar cells right now are not very efficient for a number of reasons, but that's one of them. We'd like to use this nanoparticle assembly for something called a dye-sensitized solar cell to try and make that process better. But the question we have to answer before we can get to that is, does this even work? Can we get energy to transfer across a gold nanorod? And so the way the physics behind it and how it should work is, we would want one dye, and I've shown a couple here. These are proprietary dyes from Thermo Fisher, Lexaflor 610 and Lexaflor 790. The dotted lines represent absorption curves. The glowy lines represent emission curves. So Lexaflor 610 absorbs maximally here at the dotted line, emits maximally here at the solid line. We would overlap the emission of Lexaflor 610 with the absorption of my nanorod, that's that dotted line in green here, which then also overlaps with the absorption of Lexaflor 790. So we could put in energy at Lexaflor 610, meaning shine a laser on this thing with 610, and see if we get Lexaflor 790 to respond, to kick out light. And that's how we would know that these are transferring energy. Because you can see that the overlap between Lexaflor 610 and Lexaflor 790 right here is pretty low. And so we would expect a low conversion of energy, what we call force to resonance energy transfer. Shouldn't work out very well between these two dyes. They don't overlap very well. So even if they're super close, they shouldn't transfer light very well. But the hope is if a gold nanorod's present, that we could get the light to transfer between these two. So that's what we're shooting for. So then the question is, how does one build one of these systems, right? We have nanoparticles. These things are incredibly small. I should have brought this up at the beginning, but when I say nanoparticles, if you look at an old penny, on the back of that penny, there's the Lincoln Memorial. So the new pennies have like a shield, and this example doesn't work for that. Old pennies have the Lincoln Memorial. If you envision an eyelash on Abraham Lincoln in that memorial, that's about 100 nanometers. So my particles are half that size, really, really small. And so I can't just like set them on the table and use tweezers to align them the way that I want. The question that we have that we're trying to explore here is, all right, we can make the particles, how do we organize them so that they stack up the way that we want them to? And the answer that we've been shooting for here is to use origami. To use DNA origami. This was a technique developed in 2006, so it's not that old, but it is, when it was first put out, it was a Nature paper by this guy, Paul Rodeman. He's actually a computer engineer. He was looking at DNA and was like, that's code. It's just letters that are arranged to deliver information. I wonder if I could use that code to build structures. And so he actually developed a computer program that said, hey, if you take a long piece of DNA and you fold it into a shape and throw other pieces of DNA, you can get them to arrange and create that shape a bunch. And so here's, and I'll show you a video that'll make this hopefully a little bit easier. But you can see the kind of theoretical model here. So you take a long rubber band and you fold it up into a smiley face. That's what the computer says it should look like. And then these are atomic force microscope images. These are real samples. And it's kind of creepy, right? There's all these like smiley faces floating around in the bottom down here. So the system works really quite nicely, taking advantage of DNA in a way that it really wasn't intended to be used. Right? DNA is an information source, and Dr. Rodeman said, you know, let's use it as a building block. So here's a video to highlight what this looks like. What this is going to show is a long, what we call scaffold strand of DNA. So that's something we get from nature typically. It's a genome. Often we'll use viral genomes. And then you're going to see a bunch of colored strands that come in here. And what we've done is use the computer program to say, take that giant scaffold strand. I want to fold it into, I think in this case, a square. So raster it back and forth into a square. And then the colored strands are going to hold all those DNA pieces together into that strand, or into that shape. So the gray thing coming in is our scaffold strand. The color ones are the staple strands. And we would mix all these together, heat them up and cool them down. That's called annealing. And you can see how these scaffolds, or how the scaffold strand starts to take shape. We got a helice or a helix starting to form down here. And a staple strand starts to bind to this helix and then jumps up to that helix, or binds to this helix and jumps up to that helix. And after a while of this, you get these to all form rows of helices that are held together at these crossover points with staple strands. And that's what creates the DNA origami. That's why it's called DNA origami. You take a scaffold strand and you fold it in like origami, and then you hold it together. So it's not like origami because you put staples into it, and that's, I think, cheating in the origami business. But that's what holds all these things together. The thing that's really, so Dr. Rodeman put this together and said, look, I can make these shapes. That's really cool. Well, the nanotech community said, wow, that is insane control on a really small scale. So this is a blow up of a particular map, a DNA map that we worked with. You don't need to be able to see any of this. I'm going to show you this object here again, where there's a rectangle here and a rectangle here, and this one's got a hole in the middle, and I'm going to overlap those. So you'll see this again. You don't need to read any of that. This is a blow up of that map. The black letters are the scaffold strand. The colored letters are the staple strands. And so what you can envision is that this black strand comes along here, envision it's connected, goes up here, connected, goes over here, back and forth. And so what you do is you say, all right, my scaffold strand has a CTC on this side, and then an AAG on that side. So I'm going to put in a staple strand that has GAG here and TTC there, so that it'll bind from this helix to that helix and hold them together, and so on and so forth. You do this thousands of times across the object. You can order all of these strands from a company. So each one of these are about 20 to 30 bases. And you can literally go to a company and type them in and order up your DNA strands that you want to get. The company has farms of nucleic acid synthetic bots that basically pump these things out. Actually, the company that I use is making a lot of COVID tests right now, so they're being a little slower on deliveries, but I'll give them some room there. And then you mix all these together. Well, so it works. One, that just is mind-blowing that you can make these cute little designs all over the place. But the other part that's really nice too is that everywhere there's a base, everywhere you see a colored letter, which you may notice there's colored letters everywhere, I can functionalize that. So I can say AATAG. All right, now on that guanine, I want there to be a thiol hanging off, or I want there to be an extra piece of DNA hanging off. And so I can fine-tune control on a sub-nanometer scale where things are located on this object. And that'll allow me to build up a scaffold. And I kind of got ahead of myself here. I should have put this up a little bit earlier on atomic force microscopes. I'll leave that alone for now. If you're curious how these images come together, I can talk about that, but I won't get too far apart here. This is that object that I was just showing you where you've got these two rectangles. The rectangles are sitting side by side, but actually they fold on top of each other. So you can oversimplify them to a rectangle that's solid and a rectangle with a hole in the middle. When you rotate it, you get this plus sign. And since there's a hole here, this area that's darker on each side looks like an equal sign. It's twice as thick of DNA. And the way we image these, you measure height off a surface with this atomic force microscope image. And so that's why you see these These areas that are lighter, they're taller off the object. And so you can see how fine-tuned of control you get from this. These little plus signs that are everywhere, the equal sign is the area that's doubly thick with DNA. And actually, this is a high-resolution atomic force microscope image. If you see the corrugation in here, it kind of looks like there's holes or almost like it's a chain link fence a little bit. You are seeing bundles of helices overlapping each other there. And so you're not seeing individual pieces of DNA, but kind of chunks of DNA that are stuck together. And so you can see the ends here kind of fraying like a rug. That's because there's DNA holding all this stuff together. For this particular model, we put some molecules on the end that really like to bind to a molecule called thrombin. And so they're called biotin. So we put them on either side of the equal sign here. And then when we put in thrombin, I'm sorry, not thrombin, streptavidin. When we put in streptavidin, you can see that it binds to either side where it's supposed to. So this is where the equal sign would be. Here's my little streptavidin binding area and my other streptavidin binding area. And you can see on a blow up of this, the two streptavidins have bound where they're supposed to bind. And so taking that idea forward, we want to use DNA origami because we can arrange these things so elegantly to try and arrange our nanoparticles. Instead of putting a protein here and here, we'd like to use a structure that looks a little different than this and put a silicon nanoparticle here, a gold nanoparticle here, and another silicon nanoparticle on the other side. And so that brings us back to this, right? How do we get these systems together? The overarching theme here is that we've got to do quite a bit of surface chemistry to get this to work. This is a didope silicon nanoparticle, the donor dye. Here's our gold nanorod, a didope silicon nanoparticle, the acceptor, and then some zoom ins here on some of the chemistry that's going on. So what we'd like to do is be able to conjugate all of these particles with DNA so that they can sit, this is a overly stylized version of DNA origami here along the bottom, so that I can put strands there to connect to my nanoparticles. And this is an overview slide, so let me go through them and hopefully it'll make a little bit more sense here. So you can envision here on this side of the DNA origami, I'd have staple strands that recognize this didope silicon nanoparticle. So that's the blue strand. This one's coming off the DNA, that one's coming off the silica. They would overlap and bind from Watson-Crick base pairing. And then in the middle, you've got a gold nanorod that has single-stranded DNA, let's say the red version, and red single-stranded DNA at its complement here in the middle of the nanoparticle. There's some challenges to being able to do this. So we've made gold nanoparticles pretty well, but then the next trick was to be able to put DNA on the outside. Michael Smith in my lab, who just recently graduated, but is still around for the next semester as he's applying to med schools, came up with this process for us. So what Michael was able to do for us is to take a DNA particle that we buy with this disulfide on the end. We reduce it with a molecule called TSEP. It's a phosphorus derivative, and it turns that sulfur-sulfur bond into a sulfur-hydrogen bond. That allows the sulfur then to stick to gold. Remember how much sulfur and gold love each other. They are BFFs. And that allows us to take our non-DNA coded particles and coat them in DNA. And the main thing here is that you can see the nanorods that we made after we coat them in DNA still look like nanorods. We didn't hurt them in the process. We tend to see the longitudinal plasmon shift around a little bit because we're changing the surface chemistry. And so we can also take advantage of this to do some neat things as well, studying how these come together. We know that we get the silica, we know that we get the DNA coated in DNA, that we get the gold coated in DNA, excuse me, because we can run a process called gel electrophoresis on it. And I'm sorry I'm throwing so many different chemistry things at you, but gel electrophoresis is a way that we can use electrical potential to pull DNA across a matrix. Think of it like a maze. So what we did is we put some gold nanoparticles that were covered in DNA and then some free DNA that was just thiolated but no gold nanoparticles into these wells in the top. So these are like racehorse stables. We then applied electric fields where there was a positive charge up here, a negative charge up here and a positive charge down here. DNA has an overall negative charge, and so it is attracted to the positive terminal. It's pulled through the gel this way. Things that have no charge or a positive charge would run the opposite way or stay put. And so the nanoparticles I showed you were kind of blue. You can see some of the blue that shows up here on lanes 3 and 4. These were both DNA-coated gold nanoparticles. in lane 1, that was just the DNA by itself with no gold nanoparticles present. We can put in a dye into this and see DNA specifically, something called ethidium bromide, and there's other dyes we can use as well. So you can see here, these are the DNA side of those things lighting up, showing, okay, it's actually DNA that's running through these gels. And so this particular gel electrophoresis was used to show, all right, we have DNA in here, and indeed is attached to the gold nanorods, because if it wasn't, the gold would just stay put. And so you can see here in lanes 5 is a little rough, but in lane 6, it's a little hard to see probably on your screen, but there's a little bit of purple in here, and then actually some material that's run backwards. The gold nanorods didn't come out of the lane. They just stayed put, crashed out, and then some material ran up the opposite way, which is a CTAB derivative, because there was no DNA to pull them through the lane. So when DNA is present, gold nanorods get pulled through. When DNA is not present, they don't get pulled through. And that's what I was just talking about. So the end goal here, let me back up to my slide of putting these together, would be to get our, we've got our DNA coated gold nanorods. We're working on DNA coating the silicon nanoparticles. We'd like to mix the origami and our silica particle, gold, and other silicon nanoparticle, and have them bind together. So just let them in solution bind, and the purple strands find the purple strands on the origami, the red strands find the red strands, the blue strands find the blue strands, so that our DNA acts like a scaffold and kind of prearranges them. But DNA, you may be aware, is not a great structural unit. In fact, DNA gets damaged really easily, much to the detriment of human health. If you get too much sunlight or an imbalance of acidity or temperature, or if you have free radicals in your body from smoking or breathing in car exhaust, or any number of things, oxygen actually can be quite bad for our DNA. DNA damage leads to cancer and a bunch of other things. That's when DNA's structure has been compromised. So if I'm proposing using this as a material, especially for photovoltaic devices, I do not want it to be held together by DNA. That's a bad idea, because the second it gets too hot, everything's going to fall apart. So what we'd like to do is arrange all of this with DNA and then use a surface coating, a secondary surface coating on the silica that can be triggered to bind to the gold nanoparticle. And so just a small amount of chemistry here. This is something called a thiol acetate. So it's a sulfur with this group on the end, which is an acetate. Just think of that as like a hat. It's blocking the sulfur. So gold and thiol acetates want nothing to do with each other. But when you put in this other molecule called ammonium hydroxide, or hydroxamine, you can liberate this acetate to turn into a thiol, and thiols love gold. So what we would like to do is put all our particles together, have the blue silicon nanoparticle, gold nanoparticle, red silicon nanoparticle, and DNA origami all in the solution at once, have them all link up where they're supposed to, and then feed into it this hydroxylamine to create this weld between the silica particles. Then I could take my dumbbell shape and have just silica, gold, and silica and not have to have DNA present. So then I could use that to incorporate into other systems. Yeah, so that was pretty quick. Sorry, I'm a fast talker here. So hopefully what I got across here is that gold nanoparticles are fantastic. The ancients knew about them before we did, but I think didn't get to fully appreciate everything that they could be used for. We can make them in the lab. We made all these particles at NSU. And actually, I'd also like to highlight that we are now imaging these particles at NSU with our brand new to us, transmission electron microscope. So if you ever want to come see this instrument, I'd love to give you a tour on the BA campus. And we are currently in the process, like I said, of DNA coating silicon nanoparticles that we can make these full assemblies. And once we have all of them together, that's when the real analysis of, can we tune this and change the shape of the silica and the contents of the silica to get energy to transfer from one side to the next. And so with that, I'll like to thank my group. There are a lot of folks that have come through my lab in my short time here, and the work would not be possible without it. I've had just tremendous students come through the lab. I highlighted the work of Stephen and Michael, but really there's a group effort here across these three domains of the silicon nanoparticle team, the gold nanoparticle team, and the DNA origami team. Presented all over the place. This is at NSU Undergraduate Research Day a few years back. This is at a local ACS meeting I've had students present nationally at Kerr, and we've had a good success. And you know, I'm always happy that my students are graduating and going off and doing big things, but I wish they were always still around because they've just been wonderful. So I'd like to thank my students and also NSU Faculty Research Council has provided the startup funds for this. And then we've been very fortunate to receive some pretty steady funding from Oklahoma Inbray over the last several years as well. So with that, are there any questions?
Okay, I guess let's give Dr. Green a round of virtual applause first. I mean, it's a wonderful talk. I really enjoyed it. Or yeah, whatever symbol you choose, you know.
I had the same issue when I was running the webinar. I was like, I guess I'll clap for everyone. Someone on the other side.
Yeah. Okay. We have a. Let's do what we did last time. If you have a question, please raise your hand in on the from the participants or a list. I mean, then I can then I see the hand and then I'll call you. That'll be easiest to handle. Maybe let me start from asking some questions, so maybe then the student can follow.
Sure.
I think first of all, I think this talk is really interesting, but I got some basic questions here. First of all, like starting at the very beginning, you were talking about the assembly of the gold particles, right?
Mm-hmm.
Themselves. So what is the driven force for the assembly? What kind of interactions we're talking about? Like
what kind of interactions hold the assembly together?
yes, yes.
So the sulfur gold bond is a covalent bond. It's a very strong interaction.
Okay.
Okay. So that's between the silica and the gold. Between the silica gold and DNA, those are all hydrogen bonds because it's DNA Watson-Crick base pairing. And so we have to worry about all the factors that go into hydrogen bonding, salt, temperature, acidity, all sorts of stuff can mess that up. And so those are more sensitive interactions.
So what about there's assembly of gold particles? What kind of interaction is between the gold particles? Like one gold particle, another gold.
Oh, between gold particles, they would stick together. So gold metal, if you take any metal to a zero valence state, especially at that size, they tend to link up pretty well because of the overlap of conduction valence bands. They are no band gap materials, and so they tend to fuse together pretty well. The reason that doesn't occur, let me go back to my slide up here. So you can kind of see on this slide, sorry, I'm trying to manage my screen out of people out of the way here. You can see that the particles here are not all stacked on top of each other. That's because these silicon nanoparticles have a coating. And so it's important that you coat the outside with something at all times. If the coating goes away, then the gold starts to fuse together and they're no longer they're no longer individual particles. They start to form bulk particles. So this one is a citrate coating. With the gold nanorods, it's a C-tab, that surfactant acetylene coating that stays on the outside.
So is this kind of like a maybe hydrophobic interaction for the gold particles to get together, or it's not?
Why are they crowded like this?
No, I mean just for the gold particles to get together. Is this kind of like hydrophobic? Because
no, so with citrate it would be dipole interactions.
Okay.
Like citrate's a charged molecule with
so basically like van der Waals, right? Kind of van der Waals.
No, stronger than van der Waals. Van der Waals would be the transient dipole moment. This is a permanent dipole moment of the coating.
Okay.
So the citrate has a full negative charge that would have a dipole moment with polar, with hydrogens and things in another citrate molecule. With C-tab, it forms a myocell, and so you would have some hydrophobic interactions, which are more van der Waals, but you can also have dipole forces there as well. So the good news is the coatings, they're all pretty weak intraparticle interactions. And so you want that, right? You don't want a bunch of gold nanoparticles to stick together. They look like they're stuck together here because of the way the sample was prepared. You put an aqueous suspension on a TEM grid, and then you wick it away. So as the water is wicking off the surface, it tends to kind of bunch things together. And so that's why you see that happening here. When they actually fuse together, they look like one big blob, and you can't tell them apart.
Okay, I guess anybody have any questions? Please raise your hand, and then I'll call your name, and then you can ask a question. Okay, maybe let me ask another question.
Cleared
So
everybody
basically,
off.
I'll try to maybe because my question be easier for them to ask questions. So basically, your talk kind of like there's basically two parts, right? One, the first part is about the silicon gold nanoparticles, right? This kind of a, and then the second part is about the DNA or origami, right? And then you kind of then in the end you kind of put them together, and then the ultimate goal is kind of get them together. So I guess my question is like, why do you need DNA in your game? I mean, I understand you want to make this kind of like the donor and etc. Like this kind of a connection, like the two gold particles. I mean, no, sorry. And then one rod in the middle, right? And then why do you need DNA in there?
Because if you, let me go forward here. If you look at the number of particles that are present here, we'll go to the cool one. I spent more time on that one. All right, so the number of particles that are present here, you could get a lot of iterations of this. So we have a donor particle on this side, an acceptor particle on that side, and a gold nanorod in the middle. If I mix those three particles together, the chances of this occurring this way are very low. And so you can envision there'd be at a best a blue particle here and a blue particle here, or a red particle here and a red particle there, or blue completely surrounding this, or red completely surrounding this, or multiple gold nanorods aggregating together, or blue particles aggregating together, or red, so on and so forth. There's so many opportunities for these particles to be misbehaved that it is, there'd be some percentage where we would get this, but then you have the problem of how do you clean it up? How do you select for the aggregate that you want out of it? So what we're trying to do is impose some order, and that's why I titled the talk "Engineering Nano Assemblies." If you just throw all these things in a bucket and shake the bucket, they're not going to come out linked up the way that we want them to. And there's kind of a rule with nanotech that, a principle of nanotech that I really like is anything that's in the bulk scale can be made more efficient on the nano scale. And so silicon photovoltaic cells right now are just bulk silicon at a PN junction. So positively charged silicon and negatively charged silicon stuck together. Well, on a nanoscale where the magic's really happening and really sub-nanoscale, that's not a very convenient way to do it because we're not engineering for the type of material that we're getting, which is electrons. If we can reduce down the footprint of our engineering, of our objects we're working with, we can make these processes more efficient. If we can get all the nanoparticles to act like a bulk. So if you have a bunch of nanoparticles, that doesn't mean they're all instantly better. You have to get the nanoparticles to all behave the exact same way. And when you have billions and trillions of these nanoparticles acting in concert, then you get the efficient interaction on the other side. And so that's why we would want to use DNA, is that we need this system to self-assemble. We need to encourage them to say, look, we've got all these particles, you guys line up the way that I'm telling you to. Because if I find one of these, great, but I'm not going to get any useful properties. If I find 7 billion all in a tight clump, then I can start to use this as a material that I could put into a solar cell or something like that.
So basically, DNA serves as a guideline, right, or scaffold, as you said, right?
That's
so
right.
that they can be positioned. All these other particles can be positioned at the proper positions, right? And then after that, you don't need DNA anymore, right? You can just throw it away.
That's right. Yeah. I would. I'm a chemist, and so I'm always thinking of different ways. DNA is really not my realm. I only recently got into this for my postdoc. And so I would like to build this initially not having DNA present at all. The idea of having DNA in there makes me nervous as a materials chemist because DNA is a horrible material. It falls apart way too easy. But it is good, I think, for in the lab under controlled conditions, getting these things lined up. Then we do that chemical modification to really lock these shapes in, and then we trash the DNA. All we need to do really is just heat them up a little bit, or we could pour in a little bit of salt, and the DNA would basically melt away from it. So the equivalent is like if you're building a building, right? You put up all the scaffolding. Scaffolding is not very strong, but it allows you to build the stronger structure, and then you tear away the scaffolding at the end.
Yes. Yeah, that's a very good analogy. Yeah, I think, yeah, that makes sense. Actually, can you go back a couple slides? Let's go back to the slides you have for the first slides for DNA origami. This maybe go forward. Yeah, like this one. I have one question. See, you have this for this setup, right? you have this two kind of, one is the you call the standard,
The scaffold strand.
the scaffold, which is the black ones, is that right?
Uh-huh.
And then you put them in the solution, and then the other ones will follow, right? The other part will follow. Is that right?
That's right. So the way that you would make one of these, this particular substrate that I'm showing you has one scaffold. It has like 7,000 plus bases in the scaffold, and it's one big circle, 7,000 bases, ATCG, blah, blah, blah. You mix in that, and then 250 unique staple strands, so these colors, and they're different. This green strand is not the same as that green strand, and so on.
Right.
You mix all of that soup into a tube with buffer and other things. You heat the tube up and you slowly cool it down, what we call annealing. And the staple strands find their scaffold equivalent and it builds itself.
Right.
So all you do is you just mix those in, heat them up, cool them down, and then you have a bucket full of these little plus shapes with equal signs in the middle.
But my question is, how do you fix the shape of the scaffold DNA in the first place? Do they have a fixed shape?
No, well, so they can have whatever shape you want. This particular one, that's why I was showing these two maps here. This particular one was designed to link up, that we call these a cross shape origami. So it was designed to link up across this plus sign, and we wanted to have arms that were unique on each side. So you can, the paper that this came from, which is here, if you want to read about it in Nanomaterials, was really actually designed to link these up and make a carpet of these plus signs. They would all link up. Like, if anybody's seen Guardians of the Galaxy, there's this scene where the bad guy's trying to get onto the good home world, and all the starships line up and kind of patchwork together to make this big shield. The same thing occurs here, is that all these particles would link arms and make a big quilt. And so we designed this particular system to look that way, but we could put in whatever sort of scaffold that we want. So you can see here, this was designed by a computer to raster these back and forth to make different particles. But sorry, I have students.
But my point is, like, the scaffold DNA is basically a single strand, right? The single strand DNA.
It's one large single-stranded piece of DNA.
Yeah, then the other part can assemble. But so do you just put them on like a substrate or something so that the fixed is fixed positions? The single strand?
Yes. So once it's formed, so all of this stuff happens in solution. You mix in everything in solution, and you deposit it on a surface. So we use something called mica. It's a mineral that is really nice because it's very flat. It's atomically flat, and so anything that sits on the surface, DNA is not very thick, right? Anything that sits on the surface, you can tell it's there because the background's so flat, and that immobilizes it. So that when we actually take the image using this thing called an atomic force microscope, which basically is like an atomic record needle that bounces along the surface and registers high spots, we'll run across these as they are fixed on the surface. So that's how we get these images, that they are stationary when they're made for imaging. But they're stable in solution. So all these little plus signs that you see here, they're floating around in solution, stable. So it would look, if you could see in this solution, you'd see plus signs floating around with a little bit of flexibility because DNA is not very rigid.
Yeah, I like it. It's really interesting. Yeah, I mean, it's kind of like you're manipulating stuff like nanoparticles with the help of DNA and stuff.
Yeah, when I first saw the Nature paper with the smiley faces, and he made one actually of a map of the world and it's just repeating over and over, and he had one that says DNA over and over and over. I was like, man, that is so cute. I wonder if that's good for anything. And really, the whole field has been saying that for a few years now. And then just more recently, people have actually been using these to make boxes that can deliver drugs and different structures that you can manipulate with light to actuate. There's a little robot made out of DNA origami that can be used as you shine lasers on it. So there's applications that are starting to come into the field with DNA origami, and it's amazing. It's one of those things where you're like, why didn't I think of that? That's so simple. You just match A, T, C's, and G's. And you can do this on paper. You can line it up on paper, but there's computer programs to make these for us.
Yeah. So in some sense, DNA serves as a catalyst, like kind of help this guy. I mean, probably not, but kind of like, is that right?
Yeah. So catalyst, to a chemist, catalyst is a very specific term. So it is consumed in the reaction. But yeah, the DNA allows these. The DNA is not the final product. The DNA is meant to help it get there.
Yeah. Okay.
Parker, I feel like you guys have a question. I can just tell there's something on the tip of your tongue here.
Yeah, go ahead, guys. Anybody want to ask a question? I mean, you don't need to raise your hand anymore. Just talk, okay? Okay.
What was your favorite part about doing this type of stuff, like figuring this stuff out and doing it as a project, I
Uh,
guess?
yeah, that's a good question. I was just talking to Dr. Zhang about this beforehand. Your previous speaker was talking about Richard Feynman, who is really one of my idols. And he wrote this great book called The Pleasure of Figuring Things Out that really speaks to me about what I love so much about science is that really at the end of the day, we are just little kids who are super curious and we just keep asking why until we get to an answer that satisfies us. So for me, the overarching theme of all these things that's been so nice is seeing how we can fine tune control of these structures. When we want to make gold nanorods, we can switch out small things that are put in the solution and we get a large gold nanorod or a small gold nanorod or a sphere. With the DNA origami, those are hugely gratifying to be able to see. I mean, when you see all of these plus signs that have formed, if I zoom out, they're everywhere. It's just an ocean of these things that have formed in solution. And that I made those by typing into a computer where A, T, C, and G should go, and that it actually works, and that it, that was the intent. There's something really gratifying about getting down to the individual atoms and seeing what the individual things do to change the experiment. So I find that from a material side, anytime we successfully make a material, so this image was probably the most exciting thing that I saw for origami. Honestly, for DNA, this blue color was one of the most exciting things that I saw because we knew when we made the blue color that the only way you get gold to be blue like that is to make a gold nanorod that's well defined. And so this took a long time for us just to make the gold nanorods. My students worked on that for the better part of a year. And they were getting purple solutions and red solutions, and they were just when they finally got blue, they literally like ran in like screaming like, "Dr. Gray, we got blue!" And so this is a long answer to that, but I think just the pursuit of these things, the pleasure of figuring out how these things work is hugely gratifying.
I have another question. So, do you think you would be able to take some maybe physics majors in case some of them are interested in working in your lab?
I have not, so being on the Broken Arrow campus, the reason that I have probably never met many of you is that unfortunately we don't have physics over here for reasons that, well, I won't go into it now, but I would love to. A lot of what we do is physics. We do a lot of interaction of light, of dielectric constant work, and trying to figure out how to basically exploit those interactions to get the photophysical properties we're looking for. So, if a student was motivated and wanted to try out this research, absolutely. Unfortunately, you really need to be in Broken Arrow for a lot of this work. But for the DNA work, we do a lot of computational work there, and so that could be done more remotely. But yeah, I have primarily chemistry students in my lab, but I've had plenty of cell and molecular students come through. The only reason I have not had any physics majors come through is that we just don't have many physics majors in the Broken Arrow campus.
Yeah, I guess they probably need to drive to BA like at least once or twice a week or something to work.
At least once a week, yeah, and that can be a bummer, and want everybody to be safe. So.
Yeah, but I think that nanotechnology is a really good directions for physics students. I mean, they could if they want to go grad school either in physics or chemistry or physical chemistry or something. That definitely is a good direction for them to go.
Yep. Oh, Dr. Pilcher says I think they have one physics major on the BA camp. All right. So if that physics major is here and listening, come on down. I'd love to chat with you about it. And for anybody here, honestly.
I think it's new. I mean, Dr. Higgs just mentioned that we got a new physics major. I'm not sure he's here or not, but yeah, definitely is something. At least there's an option for him. Even if he want to work with you, like in your lab, something. It'll be more convenient. Yeah.
Sure.
Great. Okay. Do we have any other questions? Anybody else want to ask a question? Okay, I think it's about time. We got 1 full hour. It's really interesting talk, and I learned a lot from it. I'm really happy like we got this opportunity. It seems like really decent. You guys doing a bunch of decent stuff there. I mean, it's a good lab for students to work on. So again, let's give our speaker another round of applause. Really, yeah. Again, really, Thank you very much for being here. And then I guess that's I'll stop recording this. And then thank you for everybody for showing up.